Cucurbitacin is any of a class of biochemical compounds that some plants, notably members of the pumpkin and gourd family, Cucurbitaceae, produce and which function as a defense against herbivores. Cucurbitacins are chemically classified as triterpenes, formally derived from cucurbitane, a triterpene hydrocarbon, specifically from the unsaturated variant cucurbit5ene, or 19,109-beta, a beta-10-alpha-lanose 5ene. They often occur as glycosides. They and their derivatives have been found in many plant families, including Brassicaceae, Cucurbitaceae, Scrophulariaceae, Begoniaceae, Elaeocarpaceae, Datiscaceae, Desfontainiaceae, Polemoniaceae, Primulaceae, Rubiaceae, Sterculiaceae, Rosaceae, and Thymelaeaceae, in some mushrooms, including Russula and Hebeloma, and even in some marine mollusks. Cucurbitacins may be a taste deterrent in plants foraged by some animals and in some edible plants preferred by humans, like cucumbers and zucchinis. In laboratory research, cucurbitacins have cytotoxic properties and are under study for their potential biological activities. The biosynthesis of cucurbitacin C has been described. Jean et al., 2014, identified 9 cucumber genes in the pathway for biosynthesis of cucurbitacin C and elucidated 4 catalytic steps. These authors also discovered the transcription factors BT, bitter leaf, and BT, bitter fruit, that regulate this pathway in leaves and fruits, respectively. The bi gene confers bitterness to the entire plant and is genetically associated with an operon-like gene cluster, similar to the gene cluster involved in phyllanol biosynthesis in Arabidopsis. Fruit bitterness requires both bi and the dominant BT, bitter fruit, gene. Non-bitterness of cultivated cucumber fruit is conferred by BT, an allele selected during domestication. Bi is a member of the oxidosqualene cyclase, OSC, gene family. Phylogenetic analysis showed that bi is the ortholog of cucurbitadienol synthase genes CPQ and squash, Cucurbitaceae. The cucurbitacins include Cucurbitacin found in some species of Cucumis pentan or Cucurbitacin or 22-hydroxy 23,24,25,26,27 pentan or cucurbit 5 and 3-1C25H40O2, white powder. Cucurbitacin B from Hemslea indicaphylla, 62Mg/72G, and other plants, e.g. Cucurbita andreana, anti-inflammatory, anti-hepatotoxic cucurbitacin B2O glucoside, from Begonia heracleofolia. 23,24-dihydrocucurbitacin B from Hemslea indicaphylla, 49 Mg/72G. 23,24-dihydrocucurbitacin B2O glucoside from roots of Picrorhiza carua. Diacetoxycurbitacin B2O glucoside from roots of Picrorhiza carua. Isocucurbitacin B, from Echinocystis fabaceae. 23,24-dihydroisocucurbitacin B3 glucoside from Wilbrandia ebracteata. 23,24-dihydro-3-apiisocucurbitacin B, from Bryonia varicosa. Pentan or cucurbitacin B or 3,7-dioxo 23,24,25,26,27 pentan or cucurbit 5 and 22-OIC acid, C25H36O4, white powder. Cucurbitacin C, from Cucumis sativus, cucumber. Cucurbitacin D, from Trichosanth scirilloi and many other plants, e.g. Cucurbita andreana, 3-apiisocucurbitacin D, from species of Elaeocarpaceae and Formium 10X. 22-deoxocucurbitacin D from Hemslea indicaphylla, 14 Mg/72G. 23,24-dihydrocucurbitacin D from Trichosanth scirilloi also from H. indicaphylla, 80 Mg/72G. 23,24-dihydroapiisocucurbitacin D, from Acanthocysseos horridus. 22-deoxocucurbitacin D from Wilbrandia ebracteata. And hydro 22-deoxo-3-apiisocucurbitacin D from Ecballia melitarium. 25-O-acetyl-2-deoxycurbitacin D, Amerinan, from Lupha amara. 2-deoxycurbitacin D, from Sloania zuliensis. Cucurbitacin E, alpha-elaterin, from roots of Wilbrandia ebracteata. Strong anti-fetid for the flea beetle, inhibits cell adhesion, also in e.g. Cucurbita andreana. 22,23-dihydrocucurbitacin E from Hemslea indicaphylla, 9 Mg/72G, and from roots of Wilbrandia ebracteata. 22,23-dihydrocucurbitacin E2 glucoside from roots of Wilbrandia ebracteata. Isocucurbitacin from Cucumis profidarum. 23,24 dihydroisocucurbitacin from Cucumis profidarum. Cucurbitacin from Elaeocarpaceae dolicostylis. Cucurbitacin 25 acetate from Helmsea graciliflora. 23,24 dihydrocucurbitacin from Helmsea amabilis. 25 acetoxy. 23,24 dihydrocucurbitacin from Helmsea amabilis, Henslicinae. 23,24 dihydrocucurbitacin glucoside from Helmsea amabilis. Cucurbitacin 2 glucoside from Helmsea amabilis. Hexanote cucurbitacin from Elaeocarpaceae dolicostylis. Persea picrosidae from Persea mexicana. Scandenicide R9 from Hemslea panacis scandens. 15 oxocucurbitacin from Cowania mexicana. 15 oxo 23,24 dihydrocucurbitacin from Cowania mexicana. Datiscacides B, D, and H from Datisca glomerata. Cucurbitacin G from roots of Wilbrandia ebracteata. 3 apiisocucurbitacin G from roots of Wilbrandia ebracteata. Cucurbitacin H, stereoisomer of cucurbitacin G from roots of Wilbrandia ebracteata. Cucurbitacin I, elatericin B from Hemslea indicaphylla, 10 mg/72g, also from Ecballium elaterium. Citrulus colocynthus, Cucurbita andreana, Dieters feeding by flea beetle. Hexanote cucurbitacin I from Ecballium elaterium. 23,24 dihydrocucurbitacin IC. Cucurbitacin L. Cacodemicides D and K from the fruits of Trichosanths tricuspidata. 11 deoxocucurbitacin from Desfontainia spinosa. Spinocides A and B from Desfontainia spinosa. 23,24 dihydro 11 deoxocucurbitacin from Desfontainia spinosa. Cucurbitacin J from Iberis amara. Cucurbitacin J 2-O-beta-glucopyranicide from Trichosanths tricuspidata. Cucurbitacin K, stereoisomer of cucurbitacin J from Iberis amara. Cucurbitacin K 2-O-beta-glucopyranicide from Trichosanths tricuspidata. Cucurbitacin L, or 23,24 dihydrocucurbitacin Bridiacide A from Bryonia dioica. Bryoamaride from Bryonia dioica. 25-O-acetyl bryoamaride from Trichosanths tricuspidata. Cacodemicides A and B from Trichosanths tricuspidata. Cucurbitacin O from Brandegia bigelovii. Cucurbitacin Q2O glucoside from Picrorhiza caruana. 16-deoxy D16 hexanote cucurbitacin O from Ecballium elaterium. D-acetylpicrocin from Picrorhiza scrophulariae. D-acetylpicrocin 2O glucoside from Picrorhiza scrophulariae. 22-deoxocucurbitacin O from Wilbrandia ebracteata. Cucurbitacin P from Brandegia bigelovii. Picrocin from Picrorhiza scrophulariae. Picrocin 2O glucoside from Picrorhiza scrophulariae. Cucurbitacin Q from Brandegia bigelovii. 23,24 dihydrodeacetyl picrocin 2O glucoside from Picrorhiza carua. Cucurbitacin Q1 from Cucumis species, actually cucurbitacin F25 acetate. Cucurbitacin R is actually 23,24 dihydrocucurbitacin D. Cucurbitacin S from Bryonia dioica. Cucurbitacin T from the fruits of Citrulus colocynthus. There are several substances that can be seen as deriving from Cucurbita 5e skeleton by loss of one of the methyl groups, 28 or 29, attached to carbon 4, often with the adjacent ring, ring A, becoming aromatic. Several other cucurbitacins have been found in plants. Constituents of the colocynth fruit and leaves, Citrulus colocynthus, include cucurbitacins. The 2-O-beta-deglucopyranicides of cucurbitacins K and L can be extracted with ethanol from fruits of Cucurbita pepo CV de angua. Pentanorcurbitacins A and B can be extracted with methanol from the stems of Mamortica carrantia. Cucurbitacins B and I, and derivatives of cucurbitacins B, D, and E, can be extracted with methanol from dried tubers of Hemslea indicaphylla. Cucurbitacins impart a bitter taste in plant foods such as cucumber, zucchini, melon, and pumpkin. Cucurbitacins are under basic research for their biological properties, including toxicity and potential pharmacological uses in development of drugs for inflammation, cancer, cardiovascular diseases, and diabetes, among others. The toxicity associated with consumption of foods high in cucurbitacins is sometimes referred to as toxic squash syndrome. In France in 2018, 2 women who ate soup made from bitter pumpkins became sick, involving nausea, vomiting, and diarrhea, and had hair loss weeks later. Another French study of poisoning from bitter squash consumption found similar acute illnesses and no deaths. The high concentration of toxin in the plants could result from cross-pollination with wild Cucurbitaceae species, or from plant growth stress due to high temperature and drought.